Good afternoon. We are ready to start our webinar now. If you want to hear the Spanish translation, please switch by clicking on the globe button at the bottom of your screen. Buenas tardes. ¿Estamos listos para comenzar nuestro webinar ahora? Si ustedes quieren escuchar la traducción en español, por favor, cambien al español haciendo clic en el globo que van a ver en la parte de abajo de su pantalla. Well, now I switch to English again, and I will be speaking English for the rest of the webinar. So, hello everyone. My name is Marisabel Caballero, and I work as Global Technical Manager for Poultry with EW Nutrition. Today, it is my great pleasure to introduce our presenters and our panelists. Our panelist is Claudio Campanelli, Technical Manager for Ruminants. Claudio?
Yes, thank you very much, Marisabel. I'm very glad to be here with you today. And I want to thank you, Johanna Fink-Gremmels, for this webinar. And of course, I want to thank you, everybody here to participate to this meeting, important meeting that we speak about endotoxin. Thank you, Marisabel.
Together, Claudio and I will assist our presenter, Professor Johanna Fink-Gremmels, in answering your questions during and after the presentation. As you know, we have a slightly different format. Professor Fink-Gremmels will quickly lead us through the essentials of endotoxins in a very short presentation, after which we will jump straight into the question and answer session. We will start by answering the questions that we have collected all through the last weeks. We thank you very much for sending them in advance. We have grouped the questions under topics and will address them specifically in those groups. Now, let me introduce you. Our presenter. It's my great pleasure to introduce Johanna Fink-Gremmels. She's a professor emeritus at Utrecht University and a distinguished expert and international consultant. Professor Fink-Gremmels, the stage is now yours.
Well, thank you very much for the kind introduction, Marisabel. And I will immediately start to share my screen with you. And
let me just interrupt a little bit because I forgot to tell you that during the webinar, you can also ask questions, of course. The questions can be asked in the question and answer panel that you will find at the bottom of your screen. And please ask them in written. Some of them might be answered simultaneously by Claudio or by me during the presentation, but we would like to leave most of them at the end of the session for our question and answer. Now, Professor Fink-Gremmels, the stage is totally yours.
Thank you very much, Marisabel. It's a great pleasure to be here, and I enjoy very much the preparation we had for this meeting because it is also a scientific exchange. Now, as mentioned before, the topic of my lecture today is LPS as one of the most prominent single inducer of inflammation, and I will briefly introduce you into the subject. And below you see here my university at Utrecht, my home university. It's one of the largest in the Netherlands and quite impressive, so you are welcome to visit me. But going to the topic, what is LPS? LPS stands for lipopolysaccharide, and you have to understand that the same compound has different names in the literature. First of all, liposaccharide is, of course, a chemical structure. LPS, the abbreviation. Here you see the chemical structure in a very schematic way, and you have the lipid part and you have the polysaccharide part. And you see here a membrane of a gram-negative bacterium, and you see that these lipid parts are coming out of it and anchored partly in the membrane and partly coming out. So this is how a gram-negative bacterium has part of its membrane to be this lipopolysaccharide. Now, as it is part of the membrane, it is also referred to as endotoxin. And perhaps most of you know the word endotoxin more because it has been used for a long time in the literature and is indeed referring to the fact that the architecture of the bacterial cell wall contains this molecule. It is in principle a chemical molecule. If you go a little bit further in the literature, you will find also the name pyrogen. And pyrogen, as a historical name, indicates that one of the major effects of these LPS is to induce fever. Fever in most animal species, and is also the most prominent sign of LPS. So the take-home message is LPS is normal, it's always in the body. In animals, LPS is released predominantly by E. coli bacteria and is a major source, these coli are the major source of LPS exposure. Now, E. coli normally live in the intestine as part of the physiological microbiome. And you all know from chickens that colibacillosis, in that case, you have too many E. coli and you get a total infection in the animal. Now, if we then look what LPS does, then this is a very complicated slide, even if it's a simple one, which I stole from my former PhD student. LPS induces inflammation and fever and does it by a very specific signaling pathways, which starts with the receptor, the so-called TLR4 receptor, and this will come back later on. And TLR4 receptors are found in the epithelial lining cells in the intestines. They are found on the dendritic cells, the pathogen recognition cells of the intestines, and on all kinds of immune cells like macrophages, granulocytes, but also the Kupffer cells, the resident macrophages in the liver of an animal. So these TLRs recognize it, and then by different pathways in the cell, the signal, the activation of the receptor is transduced, and the signal goes to the nucleus of a cell, and there it starts the transcription of pro-inflammatory cytokines. The major effect is the induction of the synthesis of pro-inflammatory cytokines, and also some enzymes like COX. Everybody who knows inflammation research knows that the cyclooxygenases are part of the inducers of inflammation. But with these cytokines, the information on an inflammatory response is transmitted to the entire body. Cytokines are distributed via the bloodstream to the entire body, and that means something that starts in the intestine as an inflammatory response can also induce an inflammatory response in other parts of the organism, you know, like the lungs, like the liver, and we will discuss that in a few minutes. Now, how get animals exposed to this LPS? You know, in the initial slide, I explained that LPS is part of gram-negative bacteria, so it is always there. Other routes of exposure are air and, well, the indoor environment, because LPS from the manure binds to fine dust particles and can then, you know, be distributed in the air, and animals can inhale it. So poor environmental conditions is a higher risk of inflammatory responses to LPS. LPS can be also in feed and water when the bacteria contamination is high, and then the animals get exposed via feed and water to additional amounts, sometimes very high amounts of LPS. But this is a question in principle of feed hygiene and water hygiene. So this could be prevented if we take carefully our quality controls within the animal unit. Nevertheless, the main exposure is so-called intestinal exposure from gram-negative bacteria releasing LPS within the intestines because they are part of the intestinal microbiota. In early days, it was assumed that LPS is only released when something really dramatic happens, so animals have heavy diarrhea or heavily diseased. Now we do know that LPS is shedded by this intestinal microbiota even when they multiply. But nevertheless, the total exposure to LPS is correlated to the composition of the intestinal microbiota, and a healthy microbiome, a healthy animal, has only minor amounts of LPS, and then the LPS is naturally occurring in the intestine, but it is not harmful. So we have to understand that in a normal physiological condition, the intestines are closely guided by an intestinal barrier, And these intestinal barrier, consisting of these epithelial cells, consisting of mucous layer, consisting of some molecules of defense, and of course the immune cells in the submucosal layer, is a very, very tightly closed environment. So LPS, which would be here together with the bacteria, can never go into these deeper parts and never reach the bloodstream. So in a healthy animal, LPS is not a real problem. It can occur in the intestine. Nature has provided a defense mechanism to prevent that it starts an inflammatory response. So what we have to take home here as a message, in a healthy gut, the intestinal barrier prevents LPS to reach submucosal structure and systemic blood circulation. So nothing happens. The animal remains healthy. The animal has a normal body temperature, normal appetite, and normal feed conversion. The situation changes under condition of stress. And daily challenges for the intestinal barrier are physiological stresses, environmental stresses, and of course pathogens. But I will focus very much on this part because this can occur in every animal at every moment. Typical physiological stresses is that we change feed composition according to the age groups. Hatching, the young chicken has to come out of the egg, is a stress for the animal. If we would go to pigs and piglets, we would have weaning and regrouping as major stress factors in the life of a pig. In a calf, it's rumen development, and in fish, particularly salmon, it's smoltification processes. These are stresses that are part of the normal life cycle of an animal and cannot be prevented. Nevertheless, these are the moments where there is a high risk that the gut barrier will be affected and will not function entirely correct and fulfill not the barrier function against LPS. Of course, we have to mention also environmental stresses. And one of the biggest stresses we have at the moment with worldwide increase in prevalence is heat stress. And heat stress, where the animal are exposed to higher temperatures, will bring during the period of high temperature during daytime that the intestines get not enough of the bloodstream because the animals are cooling down themselves, and we have an intestinal hypoxia, so less of oxygen in the intestine, less of blood flow in the intestine. And this, of course, is also a stress factor to this tissue defense mechanism. And even feed and water deprivation can be clearly measured as an impairment of the intestinal barrier function. Now, I said we are not discussing pathogens, but we have to discuss microbial toxins. And microbial toxins are, of course, these endotoxins I mentioned, but also clostridial toxins. These are exotoxins. And you know them all. In poultry, it is very important in terms of necrotic enteritis, Clostridium perfringens as a confounding factor. And we have the natural toxins in forms of mycotoxins. And mycotoxins, the most investigated one in terms of gut health function and barrier function is, of course, deoxynivalenol, DUN, that even at very, very low concentration is affecting these tight junctions between epithelial cells. All these components affect barrier integrity and may cause a leaky gut syndrome. And the word leaky gut syndrome is used to describe collectively a number of events that occur when the intestinal barrier is disturbed by one of these stresses. LPS and even bacteria can reach the bloodstream. And this is a big problem we do have. Bacteria reaching the bloodstream, you know, from colibacillosis. Then this happened. That is the worst case scenario, that through a completely disturbed epithelial cells, bacteria can enter the bloodstream. This happens in poultry very often, but also think about salmonellosis, think about other pathogens that can result in septicemia and in distribution of bacteria in the bloodstream. The milder form is that only the tight junctions, these little proteins that really pull the cells tightly together, that they are disturbed. And this is typically effect of mycotoxins at very low concentration. Then you see that only some molecules, antigens, pass the intestinal barrier, and this would be LPS. If LPS reaches as a complex bind to LPS binding protein, the circulation, it will affect other biological barriers like the lung, we see particularly in pigs, we see dramatic effects on the lung and also in calves. Also the blood-brain barrier, which we see later on in a fibrillar response in the animal, so with elevated body temperature. It will cause malabsorption, so a decreased usability of nutrients, and nutritional deficiencies, because the absorption process, for example, for minerals like calcium is disturbed, selenium is disturbed, but also the amino acid transport is disturbed. So you will have a general malabsorption syndrome. And this whole leaky gut syndrome is clinically relevant because it causes a local and systemic inflammatory response and elevation of body temperature. This elevation can be mild, okay, then you might miss it even by a clinical inspection, but these effects can be so dramatic that even shock can occur and the animal spontaneously die. Now, do we have a solution? Yes, of course we have one, but in principle, this is the next presentation you can expect from EW Nutrition. And this brings me at the moment to the end of my story. And I forgot a little bit to tell you something about heat stress, but I think, Marie-Isabelle, that comes back in the questions and answers anyway.
Exactly. Well, thank you very much, Professor Fink-Gremmels, for your short presentation. Now we will move to the question and answer session. Like mentioned before, we will start with the questions that were sent by you in the previous weeks. And we will alternate in answering your questions. As I said, similar questions were grouped together, but generally speaking, if you ask a question, chances are that you will be getting an answer. From where we receive questions? Well, we receive questions from all around the world, from America to Europe, including Asia, some questions from India, and even some questions from Australia. The first group of questions, and we got many regarding this topic, are, well, I would say are coming from Australia, from Europe, and from Latin America, and are regarding what do we see in the field when we trigger, or when endotoxins are triggered. So which events are triggering stress and endotoxins? And this is a question that I would like Professor Fink-Gremmels to answer.
Okay, thank you very much. Yeah, I could not speak Spanish, otherwise I would start with hola, buenas tardes now, but I cannot. So that are only a few words to at least demonstrate that I try my best. But my language skills are rather limited, but I can also start in Dutch if you want me to. But I think that is less prominent in our audience. Yeah. This slide you saw before. You know, we have the daily challenges for the intestinal barrier, and I now focused on the chicken, because you know what I understood from Marie-Isabelle, that many of the questions came indeed from people who are working poultry nutrition and poultry husbandry. And so it is the same slide with minor modifications, where you have the physiological stresses again, which I really find the most important part of it. And you know that in Europe, we are at the moment having new legislation on feed additives, which can be used to as physiological condition stabilizers. It is an awful word, a very complicated expression, but it simply says that these effects have been neglected in the past, and that we have now to do via feeding components, feed additives, to do something to reduce the impact on these physiological stresses. So like before, it is change in feed composition, it is hatching, early life transport. Never forget that chickens are still, in many parts of the world, transported directly after hatching to the facilities, the farm facilities. And with these transports, they have a period of starvation. We all know that the chicks have the physiological means to survive this, but for the intestine, this is still a big stress factor, because at least they had to drink water, but even that is very often not possible. So we get a disturbance of the first phase of intestinal colonization with bacteria. And this phase is physiological, but it is also part of what we are doing with feed composition in early phases of life, and feed technology. And that this would be a totally different webinar, because we also know in feed technology that this is very, very important. The environmental stresses, we have heat stress, we will explain a little bit more in detail in a few minutes. And the other parts are the same as I had before. E. coli as an infection, particularly in poultry with colibacillosis, together with Armeria, necrotic enteritis. I should mention Clostridia here again. So this is what we discussed before. So what we have to accept is that these daily challenges for the intestinal barrier cannot be avoided entirely, particularly this part cannot be entirely avoided even under optimal animal husbandry and housing conditions. Shall I continue, Marie-Isabel?
Yes. So thank you very much for this answer. And we got as well questions regarding the interaction of endotoxins and heat stress. And I think our next slides are telling a little bit about this story.
So thank you very much. So I go immediately to the next slide. This is where again, the theoretical one, LPS endotoxemia. And as I explained in principle before, the clinical signs resemble those of necrotic enteritis. And with the difference that this LPS endotoxemia can occur in all stages of life. You know, coccidiosis has a certain window where it occurs more often in chicken husbandry, and distress can occur at every stage of life, even in the adult ones. It will also occur in animals that have been treated by prevention, for example, that are coli vaccinated or have coccidiostat present in feed. So this always can happen. What happens in the animal is that if they have a leaky gut system, then these endotoxins, which come into the body, will reach the systemic circulation, but at most of the blood flow that is coming from the intestines goes into the liver. The liver is the next organ that is heavily exposed to LPS. Now, nature, normal physiology, has therefore created the set of Kupfer cells. These are residing macrophages within the liver, and they have a high capacity also to filter LPS and prevent that it goes further into the body. But their capacity is limited. So everything is based on the quantity of LPS that reaches the liver and also the systemic circulation. What we see in the animal is reduced feed utilization to the inflammatory response, drop in egg production, and an increased loss of and seriously losses in terms of the economic result. So poor growth rate, poor feed conversion rate. And to expand this a little bit, because this is, I think, what we had as a question, heat stress increases the LPS and TLR4 expression in the liver of broilers. And this was what measured here by some colleagues of us. You measure here the LPS in units per milliliter, and you see the TLR4. Remember from the slide I had in the first presentation, the TLR4 is the recognition system in the body. So the receptor that recognizes LPS. And you see that with increasing amounts, these receptors are upregulated because they have now to do so heavy work to recognize all the LPS and try to bind it. And I'm sorry, but there's a I forgot to switch off my telephone. That happens. To continue, in the poultry model, we did these experiments also with much more parameters than measured before. So it was a proof of principle. We had small little groups of animals, 15 days old broilers, and we put them under different temperature regimes. And we said, the heat stress impairs the intestinal barrier integrity. And we published a series of papers on this subject. And we measured, the heat shock proteins. We measured directly the tight junctions and adherent junctions. We measured all the inflammatory markers. And then you really can show that heat stress has a major effect on the intestinal barrier. And this is what is here also, we see the LPS go through, and we had some mechanisms why this go explained in this slide. So I think that answers major questions on the interaction between heat stress and LPS. And if everybody is interested, I can always send reprints of our work. It has been published in Portrait Science and in other avian pathology and in other international journals.
Good. Thank you very much, Professor Fink-Gremmels, for the very enlightening summary of the symptomatology, or what we can expect when we have LPS in animals, especially in poultry, and what we can see when we also have heat stress as another factor playing in this very complex situation. So now we have also some questions regarding mycotoxins. And because we can see two things here, and one of them is the effects of low levels of mycotoxins, and then the interaction of mycotoxins and endotoxins. So I think, Professor Fink-Gremmels, you have some slides about DON and LPS, exactly.
I think this is the most impressive slide to see for everybody. This is done in an experiment, and we did a series of that, giving the animals, these are pigs, giving the animals DON and LPS. The pig is the most sensitive animal species, so it was a little bit, the first target to assess what happens in pigs. And these are low levels of DON. These levels given to the animals are below the level, or around the level that is permitted in Europe. And Europe has quite a strong regulation. It is only a recommendation, but the feed industry has to comply with very low levels of DON in feed materials and finished feed. And so we studied this as these very low levels. And you see here in the liver, that LPS alone can already have a dramatic effect, because you see here several hemorrhages. But if you have the combination of DON and LPS, then you see that you have really severe bleedings here. And this really shows us the interaction. DON is affecting the intestinal barrier that is well known and well studied, and allows by this the passage of the translocation of LPS, and this gives this dramatic effect in the liver. We did this work together with colleagues from Germany, with Dr. Denneke, Professor Denneke as in his team. And again, this work has been published, so you can find it in the literature, even this picture. If you go to the next slide, I will show you also a little bit of our work on DON alone, because in principle, I was discussing this now several times, that DON also at very low concentration affects the intestinal functions. The most visible sign, next to all the parameters we use with molecular biological methods like PCRs and enzyme measurements, the simple sign, what you can see here, this is a healthy small intestine, with nicely long villi, good absorption surface, so this animal will function well, will grow well, have a good feed conversion. And you see the same animal with low amounts of DON. Again, this is DON at the level that is permissible in feed. So not intoxication. It is really the low level where you would not see many symptoms at the outside of the animal. But in the inside, in the intestine, you see already that these villi are shrinking, that they are not nicely arranged here. The absorption surface is disturbed, and the nutrient transport will be much less and not very well arranged. So you have also malnutrition syndromes, so that amino acid transporters will not function and gut motility will not function always in a proper way. And these were the small intestines, and the same pictures from the last large intestines. The villi are always a little bit shorter in the large intestines, but you see here also the difference between controls and treated animals. So I think that is very nicely to see for everybody with a naked eye, of course, under the microscope. And this is accompanied also with the transport of these, we use marker molecules that we can measure. And, the transport of these marker molecules goes up. They are marker molecules, they will hardly have any transfer from the internal side of the intestines to the bloodstream. And you see, as soon as you have DON at low concentration, it is more or less from 0.5 to 1.5 units. These are measured in units. because it's a fluorescent marker. And so it is nearly 3 times as high. And to emphasize, this is measured as low concentration. This was just below the 900 ppb that are permissible in Europe for finished feed for pigs.
And how can we translate it to poultry? I think in the next slide, we are showing this study, and this is a summary of 18 different experiments that were done with broilers. And what it is showing, well, first I have to mention that all the broilers were slaughtered at the same age. So all of these show a similar feed, so no experiments going into the feed. So, and no different feed additives. The only difference that we can see in these groups of animals that which data was taken during a little bit more than 1 year is the mycotoxin contamination. And the main 2 mycotoxins that were found, among others, were deoxynivalenol and fumonisin. What could be found here is that the higher the amount of deoxynivalenol in the feed, the higher the feed conversion rate that we will find. On the right side of the screen, you can see a graph showing this effect in a scatter graph. And it shows basically that for every additional 100 ppbs of deoxynivalenol that we have, starting more or less at 1,000, 1,200, we can expect an increase of FCR of 1 point, which is showing us really what the effects of low levels of mycotoxins can show in the field. Normally, 1 point of FCR for us is normal variation. However, when we have a little bit more mycotoxins, and in this case, deoxynivalenol, we can see that we will find this variation that we regard as normal. We could prevent it, of course, by using toxin mitigation strategies. Well, this is also to answer some of the questions around the effects of low levels of mycotoxins in production animals, mainly in broilers. Why is this happening? Well, this is happening, or this can be explained also on the same way as the swine experiments that Professor Fink-Gremmels is showing. We have an effect of these low levels of mycotoxins in the intestine of the animals, which hinders the nutrient absorption, which hinders the digestion process, or which also hinders the transporters of nutrients into the bloodstream. To continue with the session, we will then get another question to Professor Fink-Gremmels. And in this case, we will start talking about mycotoxins a little bit more. We have questions regarding also the low levels of mycotoxins, but if we need to use a toxin binder when we only find a low level of mycotoxins in the feed or in raw materials?
Yes, it's a very, very nice question, because it's a question you very often hear by people. You know, do we have to measure every feed batch and use only mycotoxins if we have a contamination, or shall we use a mycotoxin binder in general? Of course, I cannot answer this question because even mycotoxin binders cost money. So you have to consider the economic impact. But what you also have to consider is this. What we have is that mycotoxin binders we have at the moment in the market have also the ability to bind LPS. And LPS, as I mentioned before, of course, can occur in the life cycle of an animal at any stress moment. So of course, the effect is more dramatic when we have a co-exposure to mycotoxins as well. But on the other hand, consider that you might have heat stress, that you might have heat changes during the production cycle. And that means that many people start now to use a mycotoxin binder, not only to mitigate the effect of mycotoxins, but generally to stabilize the intestinal microbiota and the intestinal barrier with compounds that also prevent the absorption of LPS.
Sorry, Marisabel, can I say one thing? Just because it could be interesting in this moment, maybe to connect this part with a question that we have from.
Okay.
And because it's really important to understand that the low level of mycotoxin can be anyway connected with a synergistic effect with LPS. And one question is how the LPS can have an impact in sow productivity. And especially considering that DON, for example, is quite often present in our feed, and sow are absolutely a sensitive animal of trichotoxins, and so the relationship with LPS could be interesting. So, Johanna, if you can give us some information about the impact on the sows of LPS.
Yeah, I mean, sows, and I have not an extra slide for this, but in a normal life cycle of a sow, they very much experience a stress situation around farrowing, parturition period. And you can see that from the outside that they have the symptoms of the MMA, mastitis metritis complex. And this already indicates that the sow is not particularly healthy. What we know is that these sows suffer from stress, they suffer from obstipation, and have a higher absorption of LPS. Now, they are well-balanced animals, they are adult, so they can cope with LPS as it looks from the outside. If you would measure body temperature, the internal temperature with a good thermograph, so not a little thermometer, but the professional, you would see that these sows still have an increased body temperature. And this is a fibrile response we see particularly in pigs to LPS exposure. What you also have to consider, if a sow has LPS, she is generally depressed, so milk production will not be very good. But on the other hand, she's also excreting LPS in the milk. And I think that is one of the dangerous parts we should really, or the hazards we really should consider. LPS goes from the circulation of the sow via the placentas into the bloodstream of the piglet. And the young piglet is, of course, affected because it has an incomplete immune system. It has to adapt to the diet. It has to adapt to the environment. So first phase, microbiome built up in the first days of life. And if it's then challenged immediately with LPS, and we did these type of experiments, you see that they have in principle a very, very poor development of their intestinal barrier activity and their intestinal microbiome. What you see in these pigs is that they are slowly growing, that they suffer from diarrhea, so that is pre-weaning and post-weaning diarrhea, and that they are much more sensitive to infections, particularly bacterial infections. We run at the moment experiments with streptococcal infections in this situation, and we see that the animals become much more vulnerable. By the way, the same applies to poultry, where we have in this first phase of life also a much less functioning intestinal barrier. Again, first phase of life is always a difficult phase in the life of an animal, and we see comparable effects in the transfer of enterococci into the poultry bloodstream and lymphatic tissue, which is at the moment in Europe a big, big problem. And again, that can be related to exposure to LPS and an impaired intestinal barrier.
Thank you very much for your answer. And now we had some questions around the usefulness of toxin binders when we have LPS challenges, or when we believe we have LPS challenges. So this will be more connected to stress. And let me answer that question. We have, for example, in vitro experiments as the one that we are showing right now on the screen regarding the absorption capacity of LPS by the products that we commercialize. And well, this is something that we have done in vitro as a proof of concept. What have we done in vivo? In vivo, it is, well, at this moment, very difficult to challenge animals with LPS in the gut, not with LPS by intraperitoneal or intravenous injections that has been done experimentally for years. But there is no challenge model of LPS in the intestine. However, we have done some experiments in pigs in the moments that they are having the most challenging situations, for example, at weaning, or for example, with sows a little bit before and after farrowing and during lactation. What we have found is that, for example, the piglets from sows receiving the product have a better body weight gain and have less diarrhea during lactation, which can be connected to less challenges in general and probably also to less LPS challenges, as LPS can pass from the sow to the piglets through the milk. So this is what we can tell you about attempts to connect the in vivo stressful events to the in vitro proof of concept that we have around LPS. Good. Let's go to the next question. And the next question is also regarding products to use as toxin binders. And the question is, how can I pick the best toxin binder, or how can I know that the toxin binder I am picking is a good one? Of course, my first recommendation is to go to our products. And I think Professor Fink-Gremmels can also give you some lights in how to choose a product independently of the brand, of course.
Okay, thank you very much, Marisabel. There are some general rules how to look. First of all, you have to have this for yourself. You have to establish a diagnosis, and diagnosis in this connect means what mycotoxins are likely to occur in my feed materials and in what parts of the world I'm living. So that might change a lot, because in Europe we have lesser amounts of fumonisin. If you are looking into South America, you have high amounts of fumonisins regularly in maize products, so in corn. So this is one of the questions you have to answer yourself. And then you define your target. What is your main target? If you have mainly one mycotoxin, you can go to the most elegant products that are these enzymatic products that can really degrade mycotoxins. But you have to know that these products have one limitation, because they are directed to one mycotoxin only. If you have a mixed diet, you normally have co-exposure to different mycotoxins. That means you cover only one, and you have to consider that the rest is still available for the animal and can have detrimental effects. And this is why most of the marketed products still are combinations of various compounds. And these compounds include the mineral clays, so the bentonite-like compounds, which perfectly bind aflatoxins, but not so nicely bind very polar compounds. And that means that they are combined with yeast cell products. And the yeast cell products that are the oligomannanes that are in the internal side of a yeast cell, normally present. And to this, they form a kind of molecular network which absorbs a lot of mycotoxins like DUN, like zearalenone, ergot alkaloids. Experiments have been conducted with a large variety of mycotoxins. Binding is not a chemical binding, so it is not completely tight or irreversible, but at least it helps to transport the mycotoxins out of the animal, or at least prevent their absorption into small intestines. And this is why we use these combination products. And the other compounds that are in a combined product are, of course, some antioxidants. Some use selenium yeast, they have selenium extra, some use some plant material. So there are a variety of products in the market, and they all have their little benefits or additional effects that you can use in a special situation. What is one of the parameters we have added is now the LPS story, because LPS is considered as one of the major complicating factor in mycotoxin exposure, and this why we focused here very much on check your mycotoxin binder, whatever you want to use, also or ask whether or not the compound used as a mineral clays are also able to bind LPS, because it helps a lot. We learned this during the seminar, and it's well published, that the combination of mycotoxins and LPS exposure for the animal gives a much more dramatic effect. It is not only additive that is at higher concentration, even potentiating the adverse effect of mycotoxins. Perhaps that answers a little bit the question for the audience.
Yes, thank you very much, Professor Fink-Gremmels. Now I would like to change the topic a little bit from mycotoxins in the feed or toxins in the feed to the dangers that, for example, bedding material can pose into swine. So we can use straw or rice hulls for bedding in swine production. So how are the toxins that can be found there harming the animals?
Well, thank you, Margaret. I assume that I should answer that question. It is a wonderful. I'm very grateful to have this, because in many presentations that is entirely neglected. We also talk about feed and feed materials and mixed feed and feed components, but we always forget the bedding. And the bedding is particular in the swine industry, a major source of mycotoxin exposure, because swine here in Europe are normally housed on straw, and increasingly due to welfare parameters, it is, the swine are nicely bedded on straw. Straw can contain particularly straw from small grains, but also a straw from maize or from rice can contain quite serious amounts of mycotoxins. And this is something the animals eat that, particularly swine consumes a lot of straw. But if you then would go also to other animals like ruminants, they of course need the roughage. And there you have also roughage as a major component that comes via the diet, but also via the bedding. And both exposure routes have to be added, making a risk calculation of exposure in an individual herd. So that is, and that has partly, I think, in early days that was noticed, but we have learned that particular fusarium species have changed their behavior in the last 20 years quite a lot. So in old publications, this is never mentioned. And first of all, people have not measured this, they have not considered it, because everybody was focusing on human consumption, and we do not consume straw normally. And that is why it has been overlooked. But now when we measure, we always can find it. Particularly when we can find in small grains in the straw of these plants, made from the plants. You even find plants.
Good, thank you very much. And now we will pass to a couple of questions from the audience before we run out of time. I would like to address the first question, and this question is, how can we measure endotoxins or LPS in feed or in water? So first, let me tell you that, like Professor Fink-Gremmels mentioned, the main source of endotoxins that can cause harm for the animals is inside the animals, coming from the microbiome. When, gram-negative bacteria proliferate, die, and the intestinal barrier is disrupted and they can pass into the bloodstream. However, we can, of course, measure endotoxins in feed or in water. One of the most popular tests is the LAL test, or there is some ELISA test like EndoLISA that can measure this in laboratories. Today, to my knowledge, there is not a fast test that I can take into the field. Good. Let's
It is not. I only can confirm it. We tried quite a lot. Under field conditions, we depend on parameters we can measure in the animal.
Okay. Yes,
And
this will be the next question. And yeah, that's what I was going to ask you. Which markers can we use to test endotoxins already in the animals, or the reaction of the endotoxins in the animals?
It is very, very difficult to measure directly LPS. It can be done, but this is really a challenge also on a laboratory setting. What we normally measure to assess exposure is to measure the so-called binding protein, LPS binding protein, LBP, abbreviated, because this is essential, because when you go back to the slides, you saw that the TLR is a very complicated system. The receptor and the LPS has to be presented to the receptor. And this you can measure, because if you have a lot of LPS in the circulation, you will have also a dramatic increase of these binding protein. So this is directly correlated and therefore a good indicator of mycotoxin exposure. This can be measured quite easily in larger animal species. It's a little bit more complicated in poultry due to the fact that they have a minor change. The LPS binding protein is slightly different in the different animal species. And so the assays we use, ELISA-based assays, do not work very well in poultry at the moment. I think that will the companies will improve and have special sets also for poultry. The other thing what we measure regularly to assess exposure is, of course, in larger animals, measuring body temperature on a professional way. So really with a deep thermometer going into the animal, or we have for the research, we have small little with a thermometer on it. We can also use thermal cameras, which is now increasingly used in the swine industry, where you can see elevated temperature by a camera, by an imaging of the entire piglet, and then you see that, for example, the areas that have a not very thick skin, so behind the ears of an animal, you can very nicely make a demographic picture and compare this, because they are very accurate. And in the meanwhile, you have these cameras. Yeah, we use very, very expensive research cameras, but you have them even as an iPhone application, so people can themselves to use it. They are not so sensitive, but perhaps. And of course, we have a number of so-called biomarkers we can measure, and these are metabolites related to the inflammatory response. And this varies per animal species. So, you know, in pigs, again, we have, in principle, we have the C-reactive protein, which is a very sensitive, very elegant marker and easy to measure. But in poultry, and we have to go to alpha-haptoglobulin, or we have to go to citrulline or other markers, and we can measure markers of oxidative stress, because we always see also oxidative stress under the situation. But oxidative stress is, of course, a very unspecific marker. So to show really in experimental work, we have to go to more specific parameters. And these are markers of oxidative stress, like hemoxygenase, not only T-bars of the MDAs, the standard assays, but also it's a little bit the modern assays, like hemoxygenase expression, like heat shock protein, HSP70 expression. So there are a variety of markers in research you can use. Under field condition, it is still a challenge.
Thank you very much.
No rapid assay for the farm level at this moment.
Exactly. We will end this webinar very soon, unfortunately. We will stick around for a few minutes and give more questions the chance to be answered. So one question that we get frequently is if we can use or should use a toxin binder while medicating our animals. And I would say that yes, of course, there is, well, in principle, with most of the products, there is no negative interaction between the medication and the toxin binder. Well, both aim to do some good to the animal, and both will protect the intestinal barrier as well. However, also we need to consider that during antibiotic use, we may have an increased concentration of LPS in the gut, and this is something that can be reduced when we use the toxin binder in combination with the medication. So we will get a beneficial effect. Yes, we will.
Yes, and in principle, perhaps I may add, if you allow me, Marisabel,
Please.
Don't forget that a lot of countries now drastically reduce the use of antibiotics, particularly in poultry.
Yeah.
Also in swine, it is mandatory. We have the subject of antimicrobial resistance as a major challenge in this century because we see a dramatic increase in resistant bacteria around the world. And so we get a prudent use of antibiotics is now, I think, a mandate that is accepted on all continents. And that we are trying to reduce particularly the use of oral antibiotics. Now in poultry, this is not possible. You cannot inject antibiotics to a herd of 10,000 little birds. So there we will use water medication, and water medication normally does not interfere with the use of a toxin binder. So if the animal is seriously ill, and if you decide to have an antibiotic in your program, think twice what other parameters have to be corrected on a farm to avoid this. But sometimes it is unavoidable. But there have been very nice experiments in terms of water medication plus a mycotoxin binder, in principle, without any kind of compromising or interacting effects in a negative way. And all the mycotoxin binders have also been tested that they do not interact with coccidostatic agents. Because coccidiostates are generally given via feed. They are standard medication still in many parts, even if we also want to avoid them on a long term, but this is less successful at the moment because we have no good alternatives. But the combination of mycotoxin binders and coccidiostat has been tested, and there was no adverse effect or adverse interaction or loss of efficacy or whatever. And yeah, you know, the most well-known example for it is, of course, you know, this is, I think, what everybody was making nervous or asking this question is, there is an effect of binding of the classical mineral clay products that are used with tetracyclines. Tetracyclines is the only class of compounds, the older ones, not even doxycycline, but the older ones, like OTC, CTC, that indeed can be partly absorbed by the binder. But you have also to consider that these older tetracyclines are poorly absorbed by the animals. So the absorption rate is in swine, which has the highest absorption rate, more or less only 3 to 6%. And you would have to fast the swine with no feed for 20 hours to get a higher absorption rate. So this is also, you know, these tetracyclines are also bound to feed particles. They have been used in the past, particularly as in-feed medication to modify the microbiome. People didn't want to accept that really, but that is a major effect. And this can be, there is an interaction between the tetracyclines and the mycotoxin binders. The big but is you will not see that in vivo because, you know, the little what you lose in efficacy of the tetracycline is highly compensated by the positive effect of a mycotoxin binder. So even if there is a theoretical ground in the laboratory, an interaction that we really can show in the animal, this in terms of animal health, this has not a big impact, because then we have a kind of a synergistic effect. It's not a real synergistic effect, but on two ways, totally different pathways, finally, the gut health of the animal will improve.
Thank you very much, Professor Fink-Gremmels. We have time only for one last question, and I will leave it to you, which is, can LPS be totally removed in blood circulation, or will it accumulate in the liver over time?
If you have an intestinal barrier that is fully intact, working properly, you will have more or less no measurable. You have really minute amounts of LPS in the circulation. After the liver, you always have some LPS in the portal system, because there is not, a gut is also a living organism, so there's also a little bit of transfer from LPS from the intestines into the portal bloodstream, so the bloodstream between the intestines and the liver. And this is why the liver has this brigade, I could call it, of Kupfer cells who do the job to prevent that it goes further into the body. And these Kupfer cells are very, very powerful. That are macrophages, very powerful in engulfing LPS and thereby inactivating it. So the nature has, as usual, several mechanisms to protect the animal. So if the intestinal barrier fails for a very short moment, the liver cells can do it, the Kupfer cells. But on a long term, if that goes over a day or 2 or 3, then the system in the liver is exhausted. The Kupfer cells are not rapidly multiplying, so they cannot adapt very nicely to a situation that exists longer than a few hours or a couple of days.
Thank you very much. I know that there are still some questions unanswered on the Q&A box. I apologize for not being able to deal with all of them during this session. However, we will be happy to pick up this conversation by email, so please write us to webinar@ewnutrition.com, and all the questions will be routed to us. Now, let me give a couple of concluding remarks of this webinar. So first, I would like to address the complexity of the gut environment and how in this complexity toxins play a role by disrupting it. When we have mycotoxins can disrupt the gut barrier and let more endotoxins to pass into the bloodstream, which will be very harmful for the animals. So what should we do here? Well, we should aim to maintain the integrity of the gut barrier by all means possible. And by that, we mean first starting with biosecurity management, nutrition, and feed additives, of course. And secondly, we can lower the work for the animal or the load for the animal regarding myco and endotoxins by mitigating this risk using the right solutions. Well, thank you very much for your attention. Stay safe and goodbye for now.
Thank you. Goodbye from my side as well.
Professor Fink-Gremmels, you are mute. You are still mute. Let me see.
Now I'm unmuted. Sorry, I want to take the opportunity also to thank the participants for listening. I hope you did enjoy our seminar a little bit. And if you have questions, I'm sure that the people from EW Nutrition will pass them to me or answer them directly. So we are happy to answer any later incoming questions as well. Thank you and have a nice evening. Stay safe and bye-bye.
Adiós, hasta la próxima.
Thank you.
And I can say goeie avond. That is typical Dutch.
Oh,
Bye-bye.